Hello students, today we are going to read about reproduction in plants. We will be mostly dealing with the asexual methods of reproduction in plants. So let us begin. So before we know what is reproduction in plants, we need to know what is reproduction. The process by which the living organisms are producing more of their kind, and it is needed for the continuity and survival of the species. That process is known as reproduction. And reproduction generally happens in two types, asexual mode and the sexual mode. Also we call it the asexual reproduction and we call it the sexual reproduction. In the asexual reproduction, the production of offspring or the child or the progeny as we call happens from a single parent without the fusion of the sex cells. Generally there are two sex cells, the male and the female, so there is no fusion of the sex cells in case of asexual reproduction, and therefore it is known as asexual reproduction. There is no fusion of the sex cells. These sex cells together are called the gametes. So we have the female gamete and the male gamete. Whereas in case of the sexual reproduction, the offspring is produced by two organisms of the same species, and the one organism is a female organism and the other one is a male organism, and when their reproduction takes place we call it the sexual reproduction. In that you need to remember that there are two gametes, one of the male and one of the female. So the male gametes are generally called the sperms and the female gametes are called the eggs or the ova. And then now we go to the asexual part. As I told, I will be discussing about the asexual reproduction in plants today. Then comes the asexual reproduction in plants. So this is generally happening in the lower forms of life like the bacteria, fungi, algae. And again there are of different kinds like binary fission and multiple fission, or only we can call it fission, which are of two types, and then budding, fragmentation, spore formation and vegetative propagation. Now we will start with the first kind, that is binary fission. Bi, the term bi means two. So when one unicellular organism divides into two daughter cells and generally of roughly around of the same size, then we call it the binary fission, which happens in case of bacteria. So if I just draw a rough diagram and show you, if this is one bacteria with the nuclear material is just lying around and it breaks into two daughter cells. So one organism without any fusion of any sex cells is producing two daughter cells roughly of the same size, then we call it binary fission. Next is multiple fission. So by the name itself you can understand that binary was two, so here it is multiple. So this is generally seen during unfavorable conditions. Some organisms, they are developing a thick covering around it which they call a cyst. This inside that cyst, suppose there is one organism and they have developed a cyst around their body, and then they divide multiple times inside it. One organism was there, now it has divided into several organisms. When they get the favorable condition, they break open the cyst and many smaller organisms which can now survive individually come out, and this is called multiple fission. Here also no fusion of the gametes are happening. It's one parent giving rise to multiple cells. This happens mostly in Chlamydomonas and cholera. Next is budding. Now budding generally happens in yeast, because we are talking here of the plants. Yeast is a kind of fungus in which a small outgrowth happens. Suppose this is a yeast cell and a small outgrowth happens which grows, and after some time it will separate from the parent and it will be ready to survive by itself and it will become independent. So that method of reproduction in yeast is known as budding, where we can see a small bulb-like outgrowth called the bud. Next method is fragmentation. In this method, the body of the organism is dividing into multiple parts. This is also particularly seen in Spirogyra, which is having a filamentous body, and each part is dividing and they are able to survive individually. So they are dividing into fragments or parts, and so this method is called fragmentation. Then we go to the next method. That is spore formation. As you can see, this method is generally happening in the bread mold. If you might have seen a bread and it is kept for some time, maybe in a wet place, or it has got old and stale, you can see some white powdery things developing in them. They are nothing but the spore. So spores are produced in sporangium. So if I just draw a sporangium, it looks like this, and there are many spores inside the sporangium. This whole thing is called the sporangium, and you can see many spores developing inside it. And again, when they get the proper condition, they will just burst open and the spores will be released, and they can again grow into a complete individual. So this happens mostly in the bread mold or the rhizopus, bacteria, mosses, and some ferns. Then our next method is vegetative propagation. Now here, you might be knowing that the flower and the plant, in the plant, if we talk about, the flower is the main reproductive organ, which later forms the fruit and the seeds. But there are the other parts like roots, stems, and leaf, with which the plant can also grow a complete plant. So when the growth of a plant is happening from the vegetative parts like root, stem, or leaf, without involving any reproductive organ, then that kind of a reproduction is known as vegetative propagation. Here you need to know that there is no fusion of gametes, and there is no seed also generally formed in such kinds of plants. Then this can be classified into two types, natural and artificial. When we talk of natural vegetative propagation method, it can happen automatically or naturally. There are different methods as I have classified. And when we talk of artificial vegetative propagation, we are particularly mentioning the involvement of human beings in the propagation of the plants. So that becomes the artificial propagation. For a quick understanding of the natural method, the underground stem. First one is a tuber. As I'm showing you a potato. This is the best example of a tuber. In this you can see these parts which are called the eye, and from here a new plant can develop. Even if you keep this potato for some days, you can see some growth is happening in this part. So if you cut and plant that, you will see a new potato plant growing. So that's a tuber, a fleshy underground stem. Then we have the rhizome. The best example is a ginger, in which you can see there are the different nodes. It's again a short branched underground stem with nodes and internodes from where the buds are there, and then again it can grow from here. So that's example of rhizome. Another example of rhizome is banana. And then there is corm. Corm is like thickened round underground stem which is having a swollen base with some leaves around. Example is the flower gladulus. Also we are having the bulb. In bulb, like onion, there are some dry membranous leaves are there, the scales, and inside it is underground stem with bud and membranous leaves called the scales. This example is onion, garlic, tulips, and all those. Also by aerial stem, like in strawberry or some grasses, you will see that they can arise from the base of the stem and grow touching this soil, they can just propagate from one part of the soil to another part, and thus they are able to generate or grow themselves. And then there is the root. Some like sweet potatoes and dahlias, they are having fleshy tuberous root with some adventitious buds, and then they can propagate and separately grow in the soil. And through the leaves of bryophyllum, you might have seen the leaf which produces the different plantlets from its tips. You can see the plant coming out from each tip of this leaf, and if that falls in the moist soil, the plantlet can grow into a new plant. Then comes the artificial methods of vegetative propagation, where first one is cutting. In cutting, you might have generally seen in the rose plants that we generally cut them and propagate them. So that's a simple cutting the plant, a healthy plant, and putting it in a moist soil, and over time it will grow roots itself. Then we do layering, where we take a part or a branch of the plant and bend it and cover it with soil, and after some time it is able to develop roots, and we can cut it from the main branch and the new plant can survive again. So that's done in lemon, jasmine, and so on. Then we have a grafting, in which we generally take two parts. Upper part is called the scion, and the lower part, it is called the stalk. And when we cut it and attach it properly and tie it, after some time both the part become one. This is of the old plant and this is of the variety that we want to put or grow, and it should be of the same species. And when we put it together, we get a new grafted plant. So it may be done in rose and mango. Then there is tissue culture, which is done completely in the laboratory condition, where also we call it micro propagation, because in this a small part of the tissue is grown in a proper nutrient medium. A particular medium is provided for the plant to grow. Then it grows and forms an unorganized mass of cells, which we call callus. From there it will then slowly differentiate into its different parts, and a plantlet will be formed, and that plantlet can then be transferred to the soil for further growth. That is done in orchids and chrysanthemum. Now, after learning all these methods, you might be wondering what are the advantages and disadvantages of this method, vegetative propagation. So if you see, the advantages of vegetative propagation is that it is a faster method, because you know that we don't have to wait for the seeds. So it can be done without waiting for the seeds. Means you can use the plants which have seed, but you don't want to wait for the seeds to start growing a new plant of that kind. Then desirable character of flower and fruits can be maintained. Like you can mix and breed the new variety of your choice, or keep that same variety for whichever is producing the better flowers and fruits. Then it is also useful for the seedless plants like banana and sugarcane and all. We can propagate it easily using vegetative propagation. Then also the plant which is produced is exactly same as the parent plant. What are the disadvantages of this vegetative propagation? You don't get a new variety, because whatever is grown is kept growing again and again, which also loses the plant. The new plant also loses its vigor and strength in this method. And also sometimes, as I told you, like vegetative propagation by the aerial strawberry and the grasses, they can just spread around in one area, causing overloading, and there is generally no dispersal in these vegetative propagation methods. So with this, I hope that you are clear with different kinds of asexual reproduction. Thank you.